So today we're going to discuss the process of transcription. First we'll see the different types of RNA that is present. So we have mRNA or messenger RNA. The DNA present in the cell will be transcribed into this format, mRNA. Then we have rRNA or ribosomal RNA. It is the one involved in the protein biosynthesis and they're very stable. Then we have tRNA or transfer RNA. They constitute about 15% of the total RNA and again very stable RNA. Then we have the small RNA. The further subgroups of the small RNA is the snRNA or small nuclear RNAs. Last, we have microRNA, which alters the function of mRNA. Now let's see the process of central dogma. So central dogma of molecular biology basically involves three processes. We have replication taking place, then there's transcription and translation. So replication is basically forming a copy of the DNA that we have. It has two strands. One is a template strand, another one is a coding strand. So these two strands are very important. We have a template strand and we have a coding strand. So the template strand basically is the one which gives rise to the mRNA. So it will be complementary to the sequence of mRNA. And the coding strand is also called the anti-template or non-template strand. It will be the opposite strand and has the same sequence as that of the mRNA. Now, once this DNA is replicated, next is the process of transcription. Transcription is basically the process of formation of the mRNA. It happens inside the nucleus, as you can see. Next is the process of translation in which the proteins are formed. This happens outside the nucleus in the cytoplasm, so the proteins will be finally formed. Now, let's look at the processes of transcription. So these are the main things happening in transcription. We have initiation, elongation, termination, and post-transcriptional processing happening. So let's look at the RNA polymerase enzyme, which is a highly important enzyme. Now in mammalian cells and bacterial cells, it's different. So in mammalian cases, we have RNA type 2 or B, RNA type 1 or A, and RNA type 3 or C. Now RNA type 2 or B is the main enzyme and it's the one synthesizing your mRNAs. And this is inhibited by alpha amanitin. Now RNA type 1 or A is important as it synthesizes rRNAs. rRNAs are synthesized, and type 3 or C is involved in the synthesis of tRNAs. Now when it comes to a bacterial RNA polymerase, it has two alpha subunits, two beta subunits, one omega subunit, one sigma factor, and two zinc molecules. Now the this beta subunit will fix at the initiation site. And then we have the sigma factor which recognizes the promoter sequence. Now looking at the signals for initiation, we have promoters, we have enhancers, and we have silencers. So the promoters, depending upon whether the organism is a prokaryote or eukaryote, it's different. So for prokaryotes, it's a Tata box, and for eukaryotes, it's the Goldberg-Huggins box. Now enhancers, as the name suggests, will increase the rate of transcription and silencers will be decreasing the rate. Now let's look at the process of initiation. So these diagrams show us the process of initiation. So in initiation, the starting point of transcription, so we're talking about a bacterial system first. So in the starting point of transcription will correspond to 5' nucleotide of the DNA. Now first what happens is this as this diagram. Here the DNA helix will partially unwind and RNA polymerase is going to bind to the promoter site. So we have the RNA polymerase enzyme here. It's going to bind to the promoter site with the help of the sigma factor that we earlier mentioned. Now that is when the pre-initiation complex or PIC is being formed. Now, when the enzyme reaches the appropriate site on the gene, the first nucleotide of mRNA will attach to this initiation site and it's going to attach to the beta subunit of the RNA polymerase enzyme, and hence the initiation process is taking place. Now, finally what happens is after about 10 to 20 nucleotides are being formed. So this is a step that's happening here. So you can see the new RNA strand is being formed here. Now when about 10 to 20 nucleotides are formed, this the RNA polymerase enzyme is going to undergo conformational change. They will undergo some conformational change and they will move away from the promoter region. That process is referred to as promoter clearance. Now, if it's a mammalian system, there are three classes of transcription factors. They have basal components, co-regulators, and activators. As we earlier mentioned, the mammalian system, they have a Tata box, and that is recognized by the Tata binding protein. There are about 7 transcription factors which are present, which is together called TF2. Now moving on to the next step, which is elongation. So in the process of elongation, this RNA polymerase will move along the DNA template, as you can see, and new nucleotides are being formed. So new nucleotides will be formed according to the base pairing rule. So this new strand which is formed will be complementary to the template strand that is formed. So you can see for C here, it is G being formed here. For cytosine, it's guanine. Complementary to that, it will be formed. So it's the same sequence as read from 5' to 3' direction of the DNA strand. Now, as the RNA polymerase is moving along the DNA template, there will be rewinding of the double helix occurring. And RNA polymerase has DNA unwinding property. Another enzyme which helps in this unwinding process is topoisomerase. So topoisomerase will help in the unwinding process. Now later what happens is a transcription bubble will be formed. Now within this transcription bubble, we have the RNA polymerase enzyme, we have the DNA, and we also have the nascent RNA present. Now, the final step is a termination step. So for termination, a very important factor is a termination protein or the Rho factor. The Rho factor is a very important factor here. So that will be binding and it identifies a stop signal, as you can see here. So finally, the RNA polymerase and the Rho factor will be released and we get the mRNA strand. Now coming to post-translational processing. So in the post-translational processing, in case of now the final mRNA that is formed is the primary transcript. It's also known as heteronuclear mRNA. In case of bacteria, this mRNA is not changed and the translation process will start even before the completion of transcription occurs. Now in a mammalian system, there are various changes happening to this initial primary transcript before it forms a mature mRNA. So these are the changes which are occurring. First, we have polyadenine A tail forming. So poly A tail forming means it is at the 3' terminal, there will be a 3' terminal is polyadenylated to the nucleoplasm. So here a poly A tail will be formed at the 3' end. It's about 20 to 250 nucleotides long and it will protect the mRNA from attack by 3' exonuclease. So we can see in this diagram below that this is actually the DNA that we have. So it has exons and introns, which I'll explain now. It undergoes transcription and then we get the primary RNA transcript, and this is the process where this polyadenyl tail is being attached to the 3' end. The next step is 5' capping. As you can see here, a 5' cap is being formed. Now this 5' cap, it is formed by 7-methylguanosine phosphate. They are capped at the 5' terminal by 7-methylguanosine phosphate. Then another post-translational step that occurs is methylation. Now methylation of N6 of adenine residue and 2' OH of ribose occurs, and that's what's commonly seen. Next, removal of introns and splicing occurs. So as we saw here, a DNA has both exons and introns. Now the exons are the coding regions. These are the coding regions and introns will be the non-coding regions. Now, what happens in RNA splicing process is that these introns will be removed because they are non-coding regions. They are removed and the exons are spliced. That is, they are put together. They are combined together to form the final mature mRNA. Now, this is exported from the nucleus into the cytoplasm where it undergoes further steps of translation.